Assalamu alaikum and welcome everyone to the second part of Molecular Biology essay preparation series, Molecular Techniques. I'm Dr. Lai Abdelati and I'm really excited to share my knowledge and experiences with all of you through this series. So let's dive in together. Today we will continue in one of the most essential tools in molecular biology, next generation sequencing, specifically pyrosequencing and RNA sequencing. So welcome to the session.
Break it down small like DNA. Learning the code where secrets stay. Chromosomes dance under the light. Molecular dreams will take their flight. Molecular magic, it's in your hands. Unlock the science and take a stand. Study hard, let your passions ignite. ASCP success is within your sight.
All right. Now let's move to the question section and I will start with the pyrosequencing part. All right. So the first question we have today, what type of sequencing is the pyrosequencing classified as? Pyrosequencing is a sequencing by synthesis method, all right, where each nucleotide incorporation is detected through a light emitting reaction. Okay? Okay, the second question we have in pyrosequencing, which enzyme releases light during nucleotide incorporation? The correct answer is luciferase, all right? In pyrosequencing, when a nucleotide is added to a DNA strand, as we see in the image, it triggers a reaction that produces ATP. This ATP then interacts with the enzyme luciferase, generating a plus light. The light serves as the signal confirming the successful incorporation of nucleotide, which helps scientists determine the DNA sequences. Okay, let's move on. All right. Which molecule is detected in pyrosequencing to confirm nucleotide addition? The correct answer is light emission. Light emission is detectable signal in pyrosequencing when nucleotide is successfully incorporated into the growing DNA strands. All right, let's move on. All right, which of the following best describes the sequencing read length in pyrosequencing? Pyrosequencing typically generates moderate length read, okay, about 100 to 400 bases, making it faster than traditional single sequencing, but shorter than some next generation sequencing methods. All right. Okay, next question. What is the rule of sulfurylase in pyrosequencing? In pyrosequencing, sulfurylase convert the released pyrophosphate into ATP as we see in the image, which luciferase then uses to produce a detectable light signal. Okay? In pyrosequencing, if no light is detected after nucleotide additions, what does it indicate? pyrosequencing, the absence of light means that the added nucleotide did not match the template basis. So no incorporation happened. So the correct answer here is no nucleotide was incorporated. Okay, let's move on. All right, the next question here. Which enzyme in pyrosequencing is responsible for degrading unincorporated nucleotides? The correct answer is apirase. Apirase is used in pyrosequencing to degrade any unincorporated nucleotides and excess ATP, right? So it's resetting the system for the next nucleotide addition. Okay, the next question. What is the substrate used by luciferase to produce light in pyrosequencing? The correct answer is ATP. In pyrosequencing, when nucleotide is added to the DNA strand, it releases what? It releases pyrophosphate. This pyrophosphate is then converted into ATP. ATP, right? This ATP which serve as the fuel for the enzyme luciferase. Okay, let's move on. All right, next question here. During pyrosequencing, which nucleotide type would cause the brightness light flash if incorporated multiple times consecutively? The correct answer here is nucleotide repeated multiple times. But let me explain it. Actually, in pyrosequencing, the intensity of light depends on the number of identical nucleotides incorporated into a row. This is known as the homopolymer. All right. If multiple identical nucleotides are added consecutively, more pyrophosphate is released, leading to the stronger light signal as we see in the image here. All right. Let's move on. Okay, the question here. What advantage does biosequencing offer compared to the traditional Sanger sequencing? All right. Biosequencing is appreciated for its fast, real-time data generation without the need for electrophoresis or fluorescent dye detection. All right, let's move on. All right, and the question here, which of the following is not required in biosequencing reaction? All right, the correct answer is fluorescent dyes. Actually, fluorescent dyes are not used in biosequencing. All right, in biosequencing, detection is based on the enzymatic light production. Okay, let's move on. Okay, next question here. Which of the following sequencing methods is the most prone to errors in homopolymer regions? The correct answer is biosequencing. Okay, actually, biosequencing can struggle with the homopolymer. I mean the stretch of the same nucleotide. Why? Because measuring the amount of light to determine the exact number of nucleotide is difficult. Okay, let's move on. Please, if you found this content helpful, don't forget to like, share, and subscribe. Your support means the world to me and help me create more valuable content for you. Thank you for being part of this journey. Let's continue. All right, now we'll move to RNA sequencing part. All right, the first question here. In RNA sequencing, what is typically used to fragment RNA into smaller pieces before cDNA synthesis? Alright, in RNA sequencing, RNA fragmentation is usually done by applying heat in the presence of what? In the presence of divalent cations like magnesium or zinc. This method breaks the RNA into smaller manageable pieces needed for efficient cDNA synthesis. Alright, let's move on. Okay, next question. Which of the following is the major challenge when analyzing RNA sequencing data? The correct answer is alternative splicing. Actually, alternative splicing allows one gene to produce multiple RNA variants, which complicated RNA sequencing data analysis. Alright? So, distinguishing these splices form correctly is the major challenge in transcriptome studies. Alright? Let's move on. Alright, the next question here. In RNA sequencing library preparation, what is the role of ligated adapters? The correct answer is to allow the cDNA fragments to bind to the sequencing flow cell. Alright, as we know, adapters are a short DNA sequencing that are attached to the end of cDNA fragments, right? They are necessary because they enable the cDNA to attach the sequencing platform flow cells for amplification and sequencing. Alright, let's move on. Alright, in RNA sequencing, why is the ribosomal RNA often removed or depleted before sequencing? The correct answer, it dominates the RNA pool and reduces sequencing of messenger RNA, right? As we know, ribosomal RNA makes about 80 to 90% of the total RNA, right? If not removed, it would consume most of the sequencing reads, leaving very little room for studying the messenger RNA or other RNAs of interest. Okay, let's move on. Alright, in RNA sequencing, what is the purpose of using unique molecular identifiers? Alright, let's define what is the unique molecular identifiers. It is the random short sequences added to each cDNA molecule before amplification. What is the benefit from it? They help differentiate true biological molecules from PCR duplicates. This leads to improving data accuracy. So the correct answer here is to distinguish original RNA molecules from the PCR duplicates. Alright, let's move on. Alright, next question here. Which sequencing technology is the most commonly used for RNA sequencing today? The correct answer is Illumina sequencing. Actually, Illumina sequencing platform dominate RNA sequence because they provide high throughput, good accuracy, and cost-effective short-read sequencing, ideal for transcriptome analysis. All right, let's move on. All right, the next question here. Which of the following is not common goal for RNA sequencing experiments? All right, RNA sequencing focus on studying RNA, such as the measuring gene expression level, identifying new RNA species, and studying splicing, right? DNA mutation in option D are usually studied through the DNA sequencing, not RNA sequencing. All right, let's move on. All right, the question here. What is the main advantage of using poly A selection during RNA sequencing library preparation? The correct answer is enriches for messenger RNA. But let me explain it. Actually, poly-A selection in RNA sequencing help enriches for messenger RNA by targeting poly-A tails on mature transcripts. Alright? This method effectively remove unwanted RNA species such as ribosomal RNA and non-coding RNA, improving sequencing accuracy and focusing reads on meaningful gene expression. All right. All right, the next question here. What is the typical use to fragment RNA or cDNA during RNA sequencing library preparation? All right. RNA or cDNA is fragmented using methods like sonication or specific enzymes to produce smaller fragments that are ideal for sequencing. All right. So the correct answer here is sonication or enzymatic digestion. All right. That's all for today. I hope you enjoyed this lecture. Please don't forget to like, comment if you have any questions, share and subscribe to our channel to receive all the latest lecture. And don't forget to click to watch the previous videos. Salam.
Break it down small like DNA. Learning the code where secrets stay. Chromosomes dance under the light. Molecular dreams will take their flight. Molecular magic, it's in your hands. Unlock the science and take a stand. Study hard, let your passions ignite. ASCP success is within your sight.